Hi, this is the first of a series of interviews that we're planning to do here at SES Research, and I'm so lucky to have an amazing first interviewee, Dr. Gozde Demirer. Hopefully I said that right. And she's amazing. She sent me her kind of background, and I was like, well, that's a lot. There's a MIT chemical engineering rising star, valedictorian. There's a small stint at Stanford, and then now at University of California. By the way, aren't they like severe rivals? Like severe, right? Did you have any hesitations going to the University of California after having spent a little time in Stanford?
Oh, no, not really. Scientifically, they're both great. So
So that was not a big. So she just has so many accolades. She'll be a professor next summer, and I'm excited to have her. Dr. Guzda, thank you for coming and joining me.
thank you. I'm really happy to be here for your first interview.
Excellent. So let's just jump in. You gave me a really nice summary of some of the research that you presented. By the way, this initial interview is kind of started because of an award we've been giving for about 12 years. It's a Young Investigator Award, and in your department has benefited from that. And I think you've done a lot of the research that helped to support. Can you talk? Let's just kind of get the broad view of, well, let's learn a little bit about you first. So talk about kind of your career, like how you were coming up through research. I think when you started, CRISPR wasn't a thing, and then it became a thing, and then you were already teaching it to students by 2018, which is like really early. Talk a little bit about that and progression and in parallel with CRISPR, please.
Yeah, for sure. So I did my PhD at University of California, Berkeley with Dr. Marquita Landry. So that's when I started with this nanotechnology and plant gene editing research. My PhD was in chemical engineering, and I've always been very interested to work with nanomaterials. Started to work with nanomaterials in my undergrad. And yeah, as you mentioned, when I was an undergrad, CRISPR didn't exist. So we were using nanomaterials to deliver drugs in mammalian cells. But when I started my PhD, I realized that there's so much that nanomaterials can offer in the plant and agriculture field as well. So it's been very, it didn't get much attention from chemical engineers or material scientists to use these materials to improve the plants rather than human health. So that's what really intrigued me to get into this research and use carbon nanomaterials for plants. When we first started, the CRISPR was then becoming a very popular area. So this is around 2014 and 15. So we were wondering if we could do this gene editing in plants using nanomaterials to deliver the needed cargoes. So that's what I did during my PhD. We developed this system using carbon nanotubes to deliver DNA, plasmids, and genes into plants. And when we were first developing it, we wanted to make sure it works. So we delivered reporter proteins like fluorescent proteins. But now we progressed into using these for edit the genes of plants so that we can improve them for certain applications.
Okay, so kind of first things that you were doing with nanomaterials really before CRISPR was even an available technology was delivering some, say, fluorescent payloads. And I know that's something that happens often just because it's easy to see that it worked.
Yeah, exactly. And it's hard to do two things that are new at the same time, right? So if you do nanomaterials and CRISPR and things don't work, you don't know which one is the problem. So you start with an easy step of something that should work using your nanomaterials. And once you optimize all the conditions, then you can move on to doing harder things like CRISPR.
Okay, very cool. So as you're doing this, like, for me, chemical engineers, so my education is mechanical engineer. Chemical engineers are those people who work harder than us to get at least their undergraduate degree. And it's not my first thought that a chemical engineer gets into kind of it in hindsight makes perfect sense, but gets into kind of plant genetic modifications. Is that a normal path, or did you kind of go down a path that's so significantly different than a lot of chemical engineers?
I feel like nowadays the breadth of work that the chemical engineers do is really amazing. You could find chemical engineers in a bunch of fields involving biology, not so much agriculture, but I think it's changing. So I wouldn't say it was very common, but I think it's becoming more and more common. So now maybe you know, like in most of the chemical engineering departments have changed their names in the last decade to chemical and biological engineering or biomolecular engineering. So there's a lot of emphasis under chemical engineering that involves biology. It's because we learn in chemical engineering classes the principles can be applied to study and engineer biology as well. So with that knowledge, I think we provide a little bit of a different perspective than a plant biologist. And I think it's most useful when these people come together from different disciplines to carry out these projects that have many portions from different fields. So I think we are bringing a lot of useful skills, and I hope to be involved with collaborative projects, I think, in the future as well.
Was there something, because there was some note that I saw that you had kind of sent me that gave me the impression that delivering value and maybe worldwide value meant a lot to you. So that was one of the things that attracted you to this. Can you talk a little bit about that? Was something from growing up, or what really did draw you to this?
Yeah, I think that's what motivates me. What I develop in my research, I want it to be useful and helpful to humanity. And the problem I'm focusing here is mostly food security now, and it's a big problem. So as we know, the climate is changing. So right, we're getting hotter and drier seasons, and that affects the plants we can grow in certain locations. The land is becoming limited, and as well our resources for like nutrients and fertilizers. So what drew me to this project is that if I was able to engineer these plants so that they could survive under these changing conditions, to make sure we'll have enough food for the future. So yeah, that's always what like having that value of research in practical and real life, it's what always motivated me. And I feel like it's kind of time sensitive as well, right? Like we can't wait for a very long time to fix this because of how fast the changes are happening right now. Yeah, so that's one thing. And then other thing is that when I first started, I was in the human like nanomedicine field, right? So I was in undergrad, I was using these nanoparticles for like cancer and diabetes drugs. But then what I realized when I started my PhD is that it's a more saturated field. So you have a lot of engineers and material scientists in that field already, and there weren't that many people in the agriculture and plant field. And I don't know like if it's normal, but that also motivated me to be like one of the first people in that field to start that and progress that. It felt more exciting to be one of the few people rather than being in a very crowded field. So that was the other thing that motivated me to go into this research.
Well, yeah, I think you can have a dramatic impact in a particular area. It makes me think of like even Fauci and how he started. He started in an arena, HIV, that nobody was interested in dealing with, and was able to really carve out a really good name for himself there, and obviously have just this amazing career. One of the things in the summary that you sent me gave me the impression that there's a different, and this may be the wrong impression, but that there's a difference or maybe even a significant difference between kind of working on genetic material in animals versus plants. Is that true? Did I get the right impression? And what is that difference if I got the right impression?
I guess that depends, like if you're asking technically, scientifically, or in terms of like regulation. I guess I could touch base on.
Yeah, touch on both of those. Yeah.
Okay. So I think technically there are additional challenges working with plant material. And I would say one of the biggest challenges is the cell wall. So that's an additional piece that plant cells have around their cell membrane. And it makes it really hard to put in biomolecules or any types of molecules in those cells because the cell wall prevents most things from entering. And that's just the defense mechanisms plants have, right? They can't just run away from stuff like animals. So they just build tougher and tougher walls. So then when you want to do any genetic engineering research, first you need to be able to overcome that plant cell wall barrier. And that really what makes nanoparticles useful, because as we know, they are small, they are tough, so they have abilities to be able to enter these plant cells. So that's one of the technical challenges that's hard to solve, which we are doing a great job with nanoparticles. The other, I guess, challenge is that when we first started to modify our plants, so do genetic modification, now we call GMO, I think there was a lot of misconception or education of the public or no education of public. So now people are scared of consuming these foods that they think they are not safe, right? So now we are different. We are doing something very different. We are the way we make the plants modified and engineered is different than how we used to make them 20, 30 years ago. But it's hard to break down those misconceptions. So then we need to really focus on telling people that what we do now is safe and is needed. Otherwise, we are going to be losing most of our food product, right? So then there are those type of public and society challenges that in animals you have ethical considerations, but I feel like it's maybe a little bit more established and organized now. Many agencies regulating those. Because it's a newer field, I think there is less work happening in terms of educating public now, and it's just they have been like growing up with these misconceptions that it's sometimes hard to break as a scientist.
So you spoke a little bit about like a difference that CRISPR is bringing into it in this nanotechnology, I think combined with CRISPR. Is it the case, this is kind of my understanding, that previous GMO stuff was really about taking components from an already existing other plant or animal and incorporating it into the existing, and now we're just, that's more of a, I don't know, a butcher's knife approach. And now with CRISPR, we have this kind of laser focus and we're able to do really, really controlled things.
Yeah, that's also true, definitely. So as you said, we used to either take parts from different plant species or bacteria that we think gives plants resistance or increased abilities for like pests. And then when you used to do those, you used to also get like bacterial genes integrated into the plants or foreign genes integrated into the plant. And that's where the safety concerns and the regulations come into play. But what we can do now, we don't need to really integrate a full gene or something from bacteria. We could just go and change one base pair at a time or do very precise changes that already happen in nature, right? Through like mutations from the sun's UV light and things that already exist in nature that doesn't involve any bacterial proteins made because as we know, they could cause some immunogenic or allergenic reactions in people when you have these bacterial proteins made, right? So we can now edit very precisely just to get the change we need that's small, that's precise, without having all the drawbacks. So I think that's something we need to keep telling people that how controlled it is and how it already exists in nature through evolution.
That makes a lot of sense. Another question popped in my head, but before I get to that question, if I remember, so I think some of the fears, and this is even when you and I first started connecting about kind of genetic modifications, in my mind, and really I did a quick Google search, there are three main reasons that we currently do genetic modification. So one of them is to make things more insect repellent. Another one is to make them more pesticide repellent. And the other one, I can't remember what the other one is off the top of my head. But the challenge that kind of I saw is, well, if we're making things more insect repellent, insects are animals, insect repellent plants typically are producing things that irritate in some way the insect. And there are some theories that like gluten and lectins in general irritate humans. So maybe that's not good genetic modifications to work on. The next is pesticide resistance. And that just inherently implies, hey, we're going to use a lot more pesticides and we don't want the plants dying with pesticides or even certain types of herbicides. Can you talk about how what you're doing and what your goals are really aren't those two things that maybe we shouldn't be that interested in?
Yeah, so those are great points, and they are the earliest modifications we have started doing in plants. And I see the motivation for it, right? Because you are growing these crops and they're getting attacked by insects and you're losing all your crop. So that's usually what motivated the academia, and also like most of the initial early times, the people who were working on these modification were companies, right? Because they're making crops, they're selling seeds, so they need to improve. So that research was strictly motivated by increasing yield under these stress conditions. So that research is still happening, but as we mentioned, it's getting more precise and more controlled. So there are ways to do make plants resistant to insects or bacteria or viral diseases without changing their lectin or other things that may be problematic for humans. So now we have better approaches to do the same applications. But also what we are doing and what I'm hoping to do in my future lab is to look at other modifications of these plants. And what I mean by other, and what I mostly hope to focus on, is to improve them for changing environmental conditions. So not so much for biotic things like bacteria, insects, but for like higher temperatures, more carbon dioxide in air, or less water, less nutrients in general. So what we can do is if we were to be able to do these, grow these plants using less nutrients, for example, and mostly nitrogen. As we know, we use a lot of nitrogen chemical fertilizers, and then over-application of them leaches into the water, causes a lot of environmental and ecological problems with these algae rushes and everything. So if we were to able to grow these plants using less nutrients, then we wouldn't hopefully use any chemical fertilizers, and then you still get the same amount of plants in given land, or even less land, hopefully. So there are things we can do that only involve changes in how they regulate their genes, so that when there's nitrogen, they can use more of it, for example, or when there's no nitrogen, they can just still keep growing for a given time. So those are some applications we can do, and there are similar applications that people have been working on to make them drought resistant, right? So, for example, if there's less water availability, can they, for example, close up their gaps so they don't lose water as much? So they have these pores in their leaves where they do the gas exchange, right? So there have been gene modifications in some plants that close those gaps so they don't lose that much water when there is drought. So those don't involve expressing any proteins or anything that could be harmful to humans. So there's all these new modifications we can do to make them more resistant to environmental changes. And that's the part of the research I'm most excited about in the future.
Well, that's very cool. And I guess is there one other application that you and I haven't touched on yet, which is, hey, can we modify them? Because I think some of this is happening so that they produce ethanol better, or they actually produce other chemicals that we can use, or other byproducts that we can actually use in other aspects of our life. And are there any that you could touch on? They're like, oh, that would be really exciting, whether you're working on it or not.
So that's absolutely true. That's a whole other field that we don't even talk about yet. So what I was talking so far is to improve plants as a food source,
right,
which is of course great, but then we could use these plants as factories as well. And that has been happening for many years, even before the CRISPR field started. So one option is to use them for biofuels, bioethanol, or materials, right? Some plants already have abilities to make these very specific, like small molecules, drugs, antibodies, or leather or polymers. Huge range of materials you can make using these. So I guess a couple of things here that we want to make sure is that you don't want to use a land and resources in a place where you can grow food, for example, right? Because humans also need food. So then we need to talk about, okay, what plants we can grow in these areas we call marginal areas. So their soil or either environmental conditions are not appropriate to grow our food sources. So if we were to be able to grow these plants for materials or fuel in these regions, then it wouldn't take away from the area we have for food. So I think if we are careful in how we design these projects, I think they have huge benefits. So as you mentioned, biofuel is becoming very a big field, and we have some plants like sorghum. They already make a lot of these oil materials. So it helps to start with those plants, and engineer them to make even more, and how to extract more efficiently. And then, of course, we need to make it economically competitive with the other types of fuel we have, because no one is going to be able to use it if it's 10 times more expensive than our current fuels. So we need to make them economically. And one way to do that is that, for example, you can grow one plant and not only collect one material from it, but let's say you collect your biofuel material, but maybe you can also collect some polymers that are used in other applications, and some other materials. So from one plant, if you could diversify the materials you collect, of course, it will be more economical because you process them all at the same time. Other one with the COVID, actually, I've been seeing that people have been making vaccine molecules, proteins, antibodies in this model plant we have. It's a tobacco plant that's very easy to infect. So you can make it do a lot of things for you. So people have been using that as like these microbe or yeast. Maybe you know that, like, we create a lot of our molecules and drugs and now microbes and yeast and tissue culture, cell culture. So they have been doing similar things with plants. So I think these are very exciting research areas that I haven't personally worked myself, but I think we can use them as a sustainable way. All they need is sunlight. All they need is the sunlight and water, and then they keep making these molecules for you. We just need to be careful. We do it in an economical manner, and then also don't compete with our food production.
So you're saying that that tobacco could actually be a healthy plant at some point for
well, I guess their purpose is not to eat it, but extract it from the plant. But so instead of tobacco, if you were to use a tomato, maybe you could just eat the fruit, and then it would have the drug or the vaccine molecules in it. So yeah,
it is, you know, I mean, you're saying that, right? But that can happen, right? That's something.
Yeah.
And there's a phrase that I really like, and you'll probably appreciate this, given even in your short career, how much has happened, you know, with CRISPR technology and what a dramatic change that is from where you started. Is that the rate of change will never be slower than it is today.
Yeah, that's a great sentence.
Right.
Yeah.
So if you're the kind of person who's like uncomfortable with how quickly things are changing, I got some bad news for you. Things are going to just continue to change faster and faster. And I think it's driven by, you know, you can now be an expert in California, and then your kind of home country of Turkey, somebody in some small university there, you guys can collaborate where, you know, 25 years ago, there's no way you could have collaborated, and you're working in your own little dark hole. And now you guys have the potential to collaborate, and you know, great minds just come up with great things. You talked about, in my opinion, you know, interdisciplinary outcomes, like when you're working with a biologist and a chemist and I don't know, an herbologist, like you just end up with these amazing ideas and things, and they come out of that. So it's just so exciting to see that. One of the things that you said in your summary was that you're working on species independent stuff. And are you still, like, where is the research in terms of, are you actively working on, hey, I'm making this plant to be drought resistant, and you're trying that in a drought situation? Or are you still kind of perfecting the technology, the gene splicing technology, so that then you can like take on the next project? Or is it like a mishmash of all of that?
So I think you made a really good point, because I think that's the dilemma of every engineer. So when you develop something, you either can take that into applications and do a lot of applications and a lot of different plant species for drought, for nutrients, this and that, or you could keep improving your molecule and you could keep making better and more advanced. And then how do we know how much of our time to dedicate to each of it? So I think about that all the time. And I'm currently, so I'm doing my postdoc currently, and I'm trying to learn a bit more about plant biology and genomics and gene regulation right now. So I took a little bit of a side step here. Because I'm trained as a chemical engineer and trying to do these things in plants, I decided that, okay, it's good for me to learn a bit of the fundamentals before I go forward. So I guess I'm not doing either of it right now. I'm doing something completely different, but I think it's very important for me to have the fundamentals. But I think when I start my own lab, then we will hopefully be able to have enough funds and people to do both of those, right? So keep developing these carbon nanomaterials and other nanomaterials for abilities that we currently don't have in plants, but which is a lot. I mean, if you think about it, we still have a lot to develop. And then also the ones we developed to take them to applications in drought. And what you mentioned with the species independence is the tools we use currently are, they only work in certain plant species because they've been developed to work in either maize or rice or the things that the companies were, hello, that's my cat, that the companies were mostly interested in developing. And then most of those methods don't work. For example, say I want to work with this very small native plant from Peru that has like some medicine molecule in it. So the tools we develop with nanoparticles, because they are not bacteria, they are not pathogens or anything. If you optimize it, they have the ability to be able to work in all these plant species, hopefully. I mean, theoretically, right? So you need to tweak some properties of it. But so that's really exciting that once we have this working in one species, then we could apply it to all the other plant species for different applications. Maybe in crops, you want to use them for drought resistance, but maybe in some other plants, you want to be able to make small like drugs or medicine or things like that. So it's good to have that ability to be able to work with these plants that we haven't even thought about working before because we just didn't know what to do. So
it's kind of like all the background research. A lot of people with this new vaccine, the mRNA vaccine, a lot of people are like, well, it just came out of nowhere. And no, there's 20 to 30 years of research into mRNA processes, and you're kind of doing that in the plant arena. And is the carbon nanotube technology that you're using, do you think, is that going to go back to animal species, or are they already kind of working with animal species with that? Or is it very plant specific because of, you talked about the protective nature plants have around their cells?
So before we use the carbon nanotubes in plants, since their discovery, they've been used in the nanomedicine applications as well. So mostly for drug delivery type of applications in cells and in culture, in animal cells or mammalian cells. So I think that's still going on, and I think people will keep using carbon nanomaterials and other type of nanomaterials for those purposes. And we have just seen how important it could be all of a sudden, or what seems to be an all of a sudden for a relevant human problem. But I guess why I prefer to focus on plants is that there are so many nanotechnologies and so many tools that already work in animals, and then there weren't any in plants, right? So I think nanomaterials will always continue to be used in animals, but I think that maybe they have ability to offer more in plants because there isn't anything else in plants that is working well. So as you said, the main thing is the cell wall. So for example, I guess to clarify a little bit, when you have an animal cell, it only has a cell membrane, which is a lipid-based material. And then you could do many things to put things in it, right? You could use nanoparticles, but you could also like heat shock it, so to open up the membrane, or you could use polymers, liposomes, lipid particles, right? So many things are able to go through that cell membrane. But when you look at the cell wall, which is not a lipid anymore, it has a lot of complex polysaccharides, very big hydrocarbons. You can't all of a sudden now open that easily using just a polymer or a heat shock. You need something a little bit more controlled and precise, like nanoparticles that can pass through that cell wall. So I think the advantages, the impact that the nanomaterials bring to agriculture and plants, maybe is even greater than the impact they bring to the animal studies. So
that makes
sense. That's why I think,
So it was my understanding, and I could be wrong, my kind of focus is more on kind of fullerenes, carbon 60, 70 on up, really carbon 60 mostly, that there were some studies that carbon nanotubes had some adverse effects in animal models. And I'm thinking about the summary you sent where you're doing some sort of polymer coat of the nanotube, and that is actually sounded like more of the problem than the actual carbon nanotube. So that kind of broad question, which is, are they harmful in some situations, and have you guys figured out how to mitigate that? Do they naturally just kind of suck into the cells? Is there any magic that you're doing, and maybe that's related to the polymer? And then talk a little bit about like how that all ties together with your research.
So obviously when we develop these materials, we need to make sure they're biocompatible and not going to be toxic to plants or humans. So what we realized with carbon nanotubes is that, and in this case, so I'm talking about single-walled carbon nanotubes. So these are hollow cylinders of just one, basically graphene sheet as a cylinder. So we find that in plants, when you put them into aqueous solutions, so that could be like a salt solution or the solution that plants have in their leaves, which is mostly water. They at concentrations we use them, the carbon nanotube itself is not causing any toxicity. And how we do this is that first, of course, you look at the leaves, you check if they do photosynthesis, you look at their cells, but you can also do an RNA sequencing experiment. So basically, after you put the nanoparticles in them, you extract the RNA and see how it's expressing all these genes it has. And then you could do that for the plant you didn't modify and the plant you modified, and see if genes are expressed differently. So you want as minimal perturbation to their processes as possible. And when we do that, we don't see significant changes happening when we put carbon nanotubes. But you mentioned carbon nanotubes all of a sudden don't fly into the cells. They need to be optimized in certain parameters. And for example, one of them is the surface charge. So we found out that they enter the cells better when they are positively charged, and which works well because then the DNA you can load on because DNA is negatively charged. So it's a nice platform. So to be able to make them positively charged, because just carbon nanotube itself wouldn't be charged, we added this polymer called polyethylenamine. So it's a PEI. It's a positively charged polymer that helped to give that charge, that helped to put the DNA on it. But then turns out that it had some toxicity problems. And if you look at the animal literature, you could actually easily find that people have found earlier that this PEI polymer and polymers like that have been causing some toxicity at high concentrations. So we saw the same thing. When you increase the concentration above a certain point, then you start to change a lot of different genes expressed. You could, even if you go above a certain point, you could see the leaves are like dying and having like parts that are not doing photosynthesis as good. So with that polymer, you need to keep it under a certain concentration. I guess, like long story short, you can make sure, like, change its concentration, change your polymer, change its charge, and then make sure that it's okay to use.
Right.
So what we can do is now, and what I'll be doing in the future is not to use the polymer to give positive charge, but modify the surface of the carbon nanotube directly by adding different residues that are hopefully less toxic than that polymer, so that we can use more of it, because the nanotube itself, as I was saying, wasn't toxic. So that limits what amount of it you can use. So that's the consideration. Of course, it's good that we check this for plants, but if we're thinking in the future, like if we want to use this in agriculture, we would also need to check its this environmental stability, how long it's going to stick around in soil, for example, if it's going to be a problem for the animals there, microbes there. So that's a whole lot of other research that hasn't been really done before that we would need to do as well. So
yeah,
definitely it's super important.
Off the top of your head, am I right? And it may have been multi-wall nanotubes, maybe it wasn't single wall, but there were some detrimental effects in animals with carbon nanotubes. Off the top of your head, were you aware of some of that, or and maybe it's just because the membrane is so much easier to permeate, or something along those lines?
Yeah, I think there is literature both ways. It's interesting actually when you look publications, you see half of them saying that okay, it had like detrimental effects, and the other half say that no, this is like perfectly fine. So I guess maybe what that tells me is that I do remember seeing the papers like you are mentioning, multi-wall, single-wall carbon nanotubes, mostly inhalation problems in like mice and lung problems, right? So maybe the reason why we see two different literature on the same topic could be that every small parameter of these nanoparticles are probably very important, right? So when we compare two studies with each other, I think we are not making sure that like, okay, they are exactly the same size, same purity, same surface charge, same everything. We are not doing that. So we are comparing this study that has like maybe a little bit of a different charge with a different study in a different human cell line. And I think it hasn't been very systematic comparisons. So I think there are formulations of it that wouldn't be toxic. Maybe, but some formulation that maybe that's toxic to one cell type may not be toxic to another cell type, right? So
yeah,
so I think it needs to be more systematic in our analysis of these studies.
We kind of have that challenge in the C60 space, and as it relates to carbon nanomaterials or nanotubes specifically, I mean, it's understood that a different diameter tube, actually one can be conductive and another could be insulative, which by the way is just fascinating to have an actual nano wire.
Yeah.
And in the C60 space, we talk about there is some literature that shows C60 to be harmful, and every time we review that literature, it looks like it wasn't processed properly for the same application. And in our case, in order to process C60, you're using some polar solvents, so maybe a toluene or a hexane. Obviously, not good things to introduce into live creatures, and so that's usually what that's been written off. In fact, I tend to say, C60 is for industrial applications, and there's peer-reviewed published research that shows if it's improperly processed, it's harmful. And then we've kind of created a trademark called ESS60, which is processed for safer human consumption for that supplement. So, so yeah, it makes sense that you've got these different challenges.
And I guess what you're talking about is like the purification is very important, right? So that's what what we've been seeing as well when we buy these carbon nanotubes. I feel like each batch may come different as well, like how it's been purified, because yeah, people make carbon nanotubes and organic solutions as well most of the times. And then they use metal catalysts like nickel and copper. And obviously none of those things are the ones you want to put into humans or plants. And then if they're not purified enough or cleaned enough and you have these materials lingering in them, it's them that's causing that toxicity and not the carbon material that you're looking at. So I think we need to also make sure that each batch is cleaned and processed at the same time and not have these like metal contaminants. And I think that's a big thing in the carbon nanomaterial field, that if you're not making them in your lab, you need to trust someone else to have cleaned them and provide them. And then that could cause differences between your experiments as well. Absolutely. If you're using a different batch from a different time or a different company.
Yeah, and if they're changing their processes and not sharing that with you, that's that's a challenge. It's something. Anytime we make changes, we've always shared that with the researchers that have been getting our material. Just kind of similarly topicked. Have you thought about graphene at all? Like smaller pieces of graphene, and has that been tossed around? And is there any literature saying that might be of value, or does it need to be folded on itself in a tube shape to really provide the value?
Yeah, so that's a great question. So we kind of found that the single wall carbon nanotubes were working for us, and we kind of tried to improve that further rather than like jumping to different materials. But so one thing we were thinking, especially with the carbon dots and C60, is that it's very small if you look at its diameter, and that's great because we want them to be small so they can pass through the small pores of the cell wall. But then because we are trying to deliver like a CRISPR protein, like a Cas9 protein, that's huge. Yeah. So you need to also consider the surface area problem, right? So yes, it's very small, but then does it have enough surface area to be able to deliver this long molecule, a lot of it? So what we realized, initially we didn't plan for it, but what was great with the single wall carbon nanotube was that it's just 1 nanometer in the diameter, but then it's very long. So it could be like 500-600 nanometer, even longer, but we worked with 500 nanometer ones. So you have this huge surface area that you can keep loading big molecules, large molecules, a lot of them. And then the diameter of it would still stay under 20 nanometers, which is kind of 20 to 25 nanometer. We think it's the limit to be able to pass through these pores of the cell wall. So it was great that it was small enough in one dimension, but gave us a big surface area. And then when you think of graphene, it has a huge surface area, but then the most commonly used ones are 40 to 50 nanometers or so. So then in both dimensions, because it's square or rectangle, right? So then we haven't tested, but I feel like maybe they are going to be less able to pass through those pores given their shape. With carbon nanotubes, we sometimes call them nanoneedles, because if they come in like the correct orientation, they could just pierce through. And as you know, they're super stiff as well. So we think that helps too. So that's why it has like a lot of good properties, but I don't think they are unique. Like I think we will eventually find more nanomaterials that are able to enter the plant cells and do applications. And then this was to put them like inside the cells in the leaves, but then some applications don't need them to be inside the cells. You just maybe want them on the leaf surface or in the extracellular areas for different applications. Then you could use all these nanoparticles if they don't need to pass the cell wall, but just be there and deliver their cargoes. So there are applications, for example, of clay nanosheets. They look like graphene, but they're from clay. They're very biodegradable, good for environment, that you could spray it on the leaves, and then they deliver this small RNA molecule that protects plants from diseases, viral diseases. And those nanosheets don't need to enter the cell because RNA is a very small molecule. Once you release the RNA travels through the plant, gives the resistance, and your nanoparticle degrades over time. So it's a really good study, for example, that shows us other nanoparticles could easily be used for different applications as well.
So how much of your day is spent kind of reading, or week, maybe it's a week, is spent kind of reading latest literature? And are you getting most of your information just from colleagues who are like, oh, did you see this latest paper? Or are you the one in the office who's like up on the latest stuff? Or is it kind of everyone? What does that environment look like?
So I guess nowadays things are a little easier. You can follow people on social media. You could sign up for these notifications when there's a paper in your field that you get a notification. So it's very rare that I need to go and search things nowadays, because I either see it on Twitter, or we have like the Slack group right in the lab. Someone either posted there, or I have these Google notifications, and so it just jumps in during my day. Even when I'm not looking, I always see them, and then you need to make sure that you read them and be on top of the literature. So journal clubs help, because you need to read for the journal club. So like in most labs and universities, you have these journal club meetings. So I would say maybe I read 2 to 3 papers a week. I mean, I could do more, but that's just the time I have to do that. There's always more coming in relevant areas. But yeah, so and I guess like as academics, we get review requests from journals, and I think that's the big chunk of me being able to read papers before even it gets published, right? Because as a expert in that area, when there's a paper, for example, if there's any nanoparticle plant work or carbon nanotube plant work, gene editing work that's trying to be published, I'm lucky to be able to be as a reviewer on that paper, right? So then I see all the research. So maybe half of the time that's how I get to read the relevant literature.
Okay, so half of your kind of staying up abreast of what's going on is actually being a reviewer, a scientific reviewer. That's kind of cool. So so is that some of your motivation for being a reviewer, or is it just kind of contributing to the field?
I mean, it's both. It's good to be aware of the papers coming in your field. So that definitely helps to be aware of them. It's also a service. Like I submit papers and someone needs to review them. So it's kind of, I do it for other people. So it's something that I'm happy to do it. And it has benefits for me as well.
That's very cool. So you're using the nanotubes. Okay, I think I just want to make sure we cover everything. And more. What are some of the outstanding questions in the plant nanotechnology area? And thank you for providing that question for yourself.
Yeah. So I wanted to talk about that because sometimes when we talk about this, it sounds like we have done everything and solved every problem, but it's actually a very new field, right? So it's been less than a decade, and so there are still a lot of questions that we need to answer. And as we mentioned earlier, it's not just going to be the chemical engineers or plant biologists to answer those questions. We need a broader team. And I guess to give some examples, maybe I'll talk about technical challenges first, and then the more of a safety and regulation type of challenges. So in terms of technical challenges, we, for example, don't know what's the cargo size you can deliver, right? So like, for example, as I was telling you, there are the cell walls, there are their pores. So what if I want to send a very big protein? Is there an upper limit of DNA or proteins that we can send with these nanoparticles? So we don't know that yet. We think it gets less and less efficient as it gets bigger, but can we find a way to make sure we could also deliver large cargo? So that's a technical unknown. Other technical unknown is that so far we've been doing these gene edits in the leaves of the plants. So these are somatic cells, right? So the leaf is not going to pass its DNA into a new generation. So if you want to make a new plant from your edited plant, you need to be able to work with their germ cells or stem cells. So the cells that give rise to next generation. So we don't know yet if nanoparticles will be able to work with those cell types in plants. So that's also another technical unknown. I guess one thing we don't know is that we develop all these technologies with carbon nanotubes, but we want them to be able to integrate with other, like with the current way of doing things in like plant breeding and in companies. People already have methods to improve their plants, right? So you don't want to develop something and just be it by itself. You want them to be able to use by companies and other people and farmers and everything. So how do we integrate this nanotechnology, which is, very molecular? You need exact techniques, you need a lot of equipment to characterize them. How can we make sure plant breeding people can still use it? So that's a very big and open question, and that's something we need to find ways to, like, I guess, either educate the other people to be able to use them or make it very easy and user friendly to use it. So that's one thing. And then in terms of regulation, we touched upon a little bit, but we don't know if these nanoparticles, once you put it into one plant, is it gonna get transmitted to next generations? For example, I added this like tomato with nanoparticles, and then will people still be eating nanoparticles within like 4 generations of that fruit if it gets like new plants, new fruits? So we need to make sure that either they don't linger to next generations or they are safe to consume. And then we also don't know when you put it into soil, is it going to go to the water? Is it going to stay there forever? Is it going to cause any toxicity to the microbes and animals? So those are some of the other questions that will decide how we regulate them and what's the safety. And those are more of a public challenges, but that needs before a lot of research so that we can answer these questions to inform those regulations. So I think those are still, I mean, I think there are more, but maybe these are some of them that come to my mind when I was thinking.
Is there a project or hypothesis that you've kind of had kind of rolling in your head and haven't yet had a time to research it and may never, that you're like, I think this is probably true, but I don't have the time to pursue it, and maybe I'll have the time when you get your own lab, maybe?
Oh, let me think. There were so many ideas when I was like writing my faculty applications. I was thinking what to put in there, and I had some ideas that were kind of a little bit, I guess, risky maybe, that I don't know if this will work. Probably I shouldn't write it down now, but I wish I could try. So I had some of those ideas.
What's one of them that maybe is the farthest reaching one, that is maybe the least likely, but I'm assuming your interest in it would be because it would have a positive impact? Or is it because sometimes it's just like, I'd just like to know as a scientist, and then other times it's like, this would have a big impact and may be really hard. So that's why I want to, that's why it gets tabled, but also why you're interested.
I guess one thing that comes to my mind right now, and maybe it's not that far-fetched because I know that some people are working on it already, is to engineer the plants with the purpose of just sucking more carbon dioxide from air. And then with the plants, I mean, they already do take a lot of carbon dioxide, right? That's how they do photosynthesis, but engineering in a way that maybe they take like 10 times more, 100 times more, and then transmit that to their soil, to the soil and to their roots, and like hide it under the soil so that you capture it from the air and then you put it underground, under soil. So that helps with the carbon dioxide emissions, right? That helps with the global warming. So if you could do it not only for carbon dioxide, because people are doing it for carbon dioxide, but could you do it for other types of dangerous gases like nitric oxide? Can you get plants to capture nitric dioxide from air and then hide it somewhere in it? But then if it's toxic to us, it's also toxic to plants. How do you make sure that the plant is going to stay alive by sucking in that much toxic gases? And then how does it store it, like, without preventing it from staying alive and doing photosynthesis? And then what do you do with all that toxic material you put underground in soil? Like, what's going to happen is that when the plant dies, it's going to release all of it into soil, and then it's just a cycle. It's going to go back to animals, air. So you don't get rid of it. You just hide it somewhere for some duration of time. So how do you make sure that it's actually going to turn into something useful, right? Like maybe you use that to make a material that's not toxic anymore and that's useful to humans. I think it's not only enough to put it underground and then hide it there for maybe 10 to 15 years, but you need to find what to do with that thing you put in there. So if we were to be able to develop something that, make the plant suck all the bad things from air and turn it into something good, like a good material that we use, and then not go back to the cycle and air again. So I guess that will be a really nice and long research, but I don't know if I'll get to do it. I don't know if it's possible. Yeah.
That's cool. You mentioned a lot of scientific equipment. What's your favorite piece of scientific equipment to work with?
Oh, favorite. I know which one is the least favorite.
Well, to share that one too, that'll be interesting.
XPS, to characterize the carbon nanotubes. X-ray photoelectron spectroscopy. It works in high vacuum conditions, and it's just every time I used it, it was so stressful. So that's definitely my least favorite one.
How does that relate to a scanning electron? How's the technology relate to a scanning electron microscope?
It uses X-ray rather than, I guess, electrons.
Right. Yeah.
Different, I guess, higher energy. So it hits the molecules, and then they give this like their resonance, their peak. So then you know what you have in your sample. Do you have silica? Do you have carbon? Do you have nitrogen? So because it's very high energy, I think you need to operate it under vacuum conditions.
And
So
ultra
I guess
high
you get
vacuum,
like,
right? It's not
yeah,
just like, oh, a slight vacuum. It's ultra high
yeah.
vacuum. Yeah.
So you need to deep pressure, like vacuum your entire chamber, and then from a different chamber, you like slowly sticking your sample. It needs to be exact in XYZ coordinates to a smallest degree, otherwise it will fall into the chamber. And then what if like all of a sudden this like pressure, the thing like break or whatever, and then you are like afraid that because it has high vacuum. So I guess that was like very stressful.
And I think this is, I want to just kind of interject here, because one of the interesting concerns that some of our customers have expressed about our product is that it's nanotechnology in their body, right? Which is a fundamental thing that is a legitimate concern. The data for C60 is pretty clear at this point, but you know, long term is still ongoing research. But there is some concern of like nanobots and controlling through this nanotechnology. And I just want to, the segue here is that even with the tunneling electron microscope, which is one of the most precise visualization tools that we have available, you can barely see a buckyball, right? Like it's just barely within the realm of visibility. And so I'm just often sharing even the fact that carbon-60 is 60 atoms, and a typical cell is on the order of 100 trillion atoms. Like, just to give you scale, we're not, you know, a cell does one thing. If you're thinking human cells, it's either a good skin cell or a good liver cell or whatever, and it's doing one little tiny thing. If people think that a 60 atom molecule is been programmed to do something, then the scale is they're not aware of the scale that's going on. So so that's your least favorite equipment. Do you have a favorite? And maybe it's just like, I don't like any of them, and that's the one I like the least.
No, that's not the case. I usually like most of them other than this. I think the one I like the most is imaging. And I guess in terms of plants, we use a lot of confocal microscopy, so confocal imaging. And it's really nice because you work on things like in a test tube, you usually never see anything until the last step. And, you know, it's just a clear liquid and a black liquid and a green liquid, and then you have no idea what's happening in there. But once you put them into the plants, you could just put it under the microscope and then see actually what you have done, right? Did you express your gene? Did you modify whatever this thing you wanted to modify? And I really like it because it's like anticipation, and then you could see it clearly in the microscope, beautiful colors, and relatively easy to operate as well. No vacuum or anything needed. So I like it. I like the visual confirmation, and that's why I think I like imaging a lot.
Well, and I think, you know, humans were certainly incredibly visual. It's amazing. I just recently got like a little VR system, and to be in a VR system, it's all visual, right? Though there's sound also, and it's just impressive how encompassing just that visual piece is. And then it's also the end of your experiment. Maybe not the end, but it's like, it's a confirmation that the experiment went well. Is it most fun with, I would imagine like phosphorescence or fluorescence in general would just be the thing that would be, it's cool. Like, that worked, it's fluorescing, and that's awesome. Is that true? Or are there even other aspects that you like to see?
I mean, we look at fluorescence, but it stands for something, right? It's like either something you expressed, or now what I do in my postdoc lab is I work with roots. So we section the roots and we stain them for like for different cell types in the root. So we could see the xylem, we could see the epidermis cells. So it's really nice that you could stain them all at different colors. So you could see like how much of this cell you had, how much of it cell wall is there. So you could look at not only the false fluorescence, but you could also look at these organelles, these cell types very specifically, and see maybe how they are different between your wild type and the mutant. Maybe you see that your mutant has like less cell wall in epidermis or something like that, that you can easily look at like anatomical differences as well. Very
cool. What's the most nerve-wracking thing you've had to do in your career?
Other than XPS, I guess.
And maybe that's it.
Yeah, it's definitely in top 5. I guess like there are in PhD there are these examinations, right? You need to pass through like preliminary qualifying examinations. So we were lucky in Berkeley, we didn't have dissertation. But that meant that our other exams were more important. So I think the qualifying exam was probably, like, I was very nervous studying for it. And at the end, it wasn't that nervous. You talk about your research and you're the expert in the room anyways, but the preparation for it was really nervous because, like, I put so much effort and I love this project. I want to be able to continue, but what if, like, my committee doesn't agree? So I think that was quite nervous. And I guess I knew I always wanted to be in academia. I knew I wanted to have my own lab, but the academia is competitive. And when I was interviewing for faculty jobs, I wasn't sure if I was going to get it. And I wasn't really interested in doing anything else. So then I was like, I guess I will try until I get it. And luckily it worked out, but that process was a little bit nervous as well.
Well, just for those who are listening, so this is the valedictorian coming out with as a BS, who is expressing, like, everyone gets nervous, and I think that's a good message for everyone, because it definitely happens. And you're good, like, giving speeches, and you're obviously doing a great job in this interview, and thank you for your time. That is not for some people, like, it's just like, oh, I had to give a speech once, and that's not the case for you. That's good.
No, I like talks. I look forward to conferences every year. Like, last year was terrible because I didn't give any talks.
Yeah.
So I mean, the Zoom talks are usually not the same. I like the crowd. I like talking about the research. I like questions at the end. So yeah, that's definitely one of the positives for me, and not the nervous part.
Very good. Well, we're right at coming up on an hour, and I think we should probably drop this. I really appreciate everything. Is there anything, like, one, how could people connect with you? Because maybe something you said could foster some collaboration, and how could they? What's the best way to contact you? And is there anything in particular that you'd like to share?
Yeah, well, thank you for today. This was great conversation. Yeah, I'll be hiring a lot of students, postdocs, trainees next year. So yeah, I hope they'll reach out to me. They could find my email address on Caltech's website. I have a Twitter account. I'm sure they could find. I would be happy to talk with anyone who wants to get into this field, collaborators. I hope to. Yeah, I guess we didn't talk about that, but I love teaching and mentoring. So I hope to be in touch with young people, and hopefully they'll reach out to me.
Yeah, you've listed a whole bunch of stuff that you've done in terms of teaching and mentoring, really back to the beginning. So that's pretty awesome. I would ask if you are going to be a mean boss, but if you're interested in teaching and mentoring, then that's kind of hard to be a mean boss and also like to teach and mentor.
Yeah, hopefully not.
and I was a little distracted. So did you give, like, what's the best way to reach out to you?
Yeah, so they could find my email at Caltech's website, and they could also reach through Twitter if they want.
Okay, excellent.
Yeah.
And was there anything to wrap up?
No, thank you. Thanks a lot. This was great.
Dr. Demirer, am I saying that right?
Yeah.
I think so. Dr. Demirer, thank you so much. This was just, I'm super geeky, so I really love this stuff. And you're obviously an amazing expert in your field, and going to go on and do some more great stuff and make sure the planet is fed. And I like eating, so I appreciate that.
I guess that should be my first motivation next time someone asks. I like eating.
Yeah. All right. Take care. Thank you.
Thank you. Bye.
Let me turn